Bismillah ar-Rahman ar-Rahim. Today, inshallah, we'll start speaking about systemic pharmacology, which are the effect of drugs on different systems of the body. Today's presentation will be the first about drugs acting on the cholinergic system. This presentation will talk about cholinergic transmission. So, what are the goals of this presentation? The major goals of this presentation is to know what are the sites of cholinergic transmission. We have also to know how acetylcholine is sensitized, stored, and released. After its release, what are the type of cholinergic receptors that it stimulates, whether they are nicotinic receptors or muscarinic receptors. And we end up this presentation with the physiological inactivation of acetylcholine, how the effect of acetylcholine is ended. And first, we are going to talk about the sites of cholinergic transmitters. Acetylcholine is one of the famous neurotransmitters. Usually, by neurotransmitter, we mean a chemical substance that is sensitized, released, and stored in nerve endings. Usually, they are sensitized, stored, and released from a nerve ending. When they are released, usually they act locally on the synapse, either on postsynaptic membrane, on postsynaptic receptors, or presynaptic receptors. Of course, this will result in several types of actions as we will discuss later, inshallah. Okay? Therefore, any neurotransmitter, usually it is sensitized, stored, and released from a nerve cell, where it will be released into the synaptic cleft, where it will act on postsynaptic receptors or presynaptic receptors. This is general for most types of neurotransmitters. So what about acetylcholine? Acetylcholine acts as a neurotransmitter at three important sites in the body: centrally in brain, and on the motor somatic nerve, and on autonomic nervous system, as we will see in a few minutes. Centrally, acetylcholine is a well-known neurotransmitter where it is involved in many very important functions like control of muscle tone. Control of muscle tone. It is well known, as we will see later inshallah, that acetylcholine is in balance with dopamine in controlling muscle tone through the nigrostriatal pathway. Also, acetylcholine is said centrally in brain, in the forebrain area. It is involved in memory and cognition. Memory and cognition. And it is well known that deficiency of acetylcholine in a certain area in the forebrain will lead to Alzheimer's disease. Also, acetylcholine, they said that they are involved in pain, especially with neuropathic pain. There are other functions of acetylcholine which we will discuss later, inshallah, when we talk about drugs which act on the central nervous system. Second site of cholinergic transmission are the somatic motor nerve. Somatic motor nerve, which is the lower motor nerve which exits from the spinal cord in connection with the upper motor nerve, which comes from the cortex, from the motor area. This lower motor nerve supply motor nerve, which is known as well as somatic nerve, supplies the skeletal muscle in general. skeletal muscles. This motor nerve, which somatic motor nerve, also sensitize and releases acetylcholine. Acetylcholine, when it released, it acts on nicotinic receptor present in the motor end plate of the skeletal muscle, which will result, of course, in muscle contraction. So this is the second site of cholinergic transmission. A third important site of cholinergic transmission is in the autonomic nervous system. And there are two places where acetylcholine acts as a neurotransmitter in the autonomic nervous system. And as we know that autonomic nervous system controls viscera in general, i.e. functions which does not under conscious control, like functions of the intestine, of the heart, and so on. Autonomic nervous system acetylcholine acts at two important sites as a neurotransmitter. First, at the ganglia, autonomic ganglia. We know that the autonomic ganglia, there is the preganglionic nerve fiber, preganglionic nerve fiber, and there is the postganglionic nerve fiber, postganglionic nerve fiber, which usually supplies viscera, in general. It's the preganglionic, which exits from the spinal cord, and this is the postganglionic, which supplies viscera. Usually acetylcholine is sensitized, stored, and released from the preganglionic nerve fiber, where it will act on nicotinic receptor, which are present on the postganglionic nerve fiber, nicotinic receptors. Usually they are known as NN, indication of neuron, nicotinic receptor type N. Okay? So this is the first type of autonomic nervous system where acetylcholine acts as a neurotransmitter in the ganglia, autonomic ganglia, whether it is sympathetic or parasympathetic. Acetylcholine also acts as a neurotransmitter at the postganglionic parasympathetic nerve fiber. So if we just imagine this is the ganglia of the parasympathetic nerve fiber, which supply different parts of the internal organs, the viscera. This is the postganglionic. This is the preganglionic nerve fiber, which releases acetylcholine. This is the postganglionic parasympathetic nerve fiber, which is usually releases acetylcholine. We sensitize and release acetylcholine. Therefore, acetylcholine is sensitized and released in the postganglionic parasympathetic nerve fiber. This supplies most of the viscera, الأحشاء الداخلية, or most of the viscera, like the intestine, like the heart, like the lungs, like whatever they are. Most of the viscera are supplied by postganglionic parasympathetic nerve fiber. These viscera usually they contain muscarinic receptor. They contain muscarinic receptors. They contain muscarinic receptors. muscarinic receptors. Okay. Therefore, this is the second type of cholinergic transmission in the autonomic nervous system, where acetylcholine is the main transmitter at the postganglionic base sympathetic nerve fiber. Therefore, acetylcholine acts a very important neurotransmitter, as we have said, in brain, also on the motor nerve ending, where it is released, it will act on nicotinic receptor present on the neuromuscular junction. Also, it acts as a neurotransmitter in the autonomic ganglia, and also as a neurotransmitter at the postganglionic parasympathetic nerve fiber. Okay. Now let us talk about the synthesis, storage, and release of acetylcholine. We just imagine this is the cholinergic nerve ending. Outside the nerve ending, there is the amino acid choline. This choline is actively uptaken into the nerve terminal using specific membrane carrier together with sodium ions. Once choline is inside the nerve terminal, it will be acetylated with acetylCoA under the effect of the enzyme acetylCoA transferase to give acetylcholine. Some of the acetylcholine molecules will leak into the synapse, but most of it will be stored in specific storage vesicles. This is known as storage vesicles, حويصلات التخزين. Storage vesicles, which will store acetylcholine. Okay, very simple. Therefore, choline will be uptaken into the nerve terminal, where it will combine with acetylCoA under the effect of acetylCoA transferase, choline acetylCoA transferase, to give us acetylcholine. Acetylcholine, once it is synthesized, it will be stored in specific storage vesicles uptaken by specific uptake mechanisms into the storage vesicle where it will be stored. Okay. Now, once the action potential reaches the nerve terminal, when the excitation comes from the centrally and it reaches the nerve terminal, it will open voltage gated calcium channels. Why they are called voltage gated? Because they open only by change in the membrane potential. Once the action potential reaches the nerve terminal, it will open voltage gated calcium channel. Calcium will rush into the cytoplasm, increasing the intracellular calcium inside the cytoplasm. Once calcium enters the nerve terminal, it causes fusion of the storage vesicle with the presynaptic membrane, releasing acetylcholine molecules into the synapse. So acetylcholine molecules will be released into the synapse. Okay. Once it is released, it will act on both synaptic membrane, as we will see in a few minutes. Postsynaptic synaptic membrane, which contains either nicotinic receptor or muscarinic receptor. It depends. If it is neuromuscular junction, it will be nicotinic receptor. If it is ganglia, it will be nicotinic receptor. If it is viscera, postganglionic parasympathetic nerve fiber, it will be muscarinic receptor. Okay, now, therefore, acetylcholine will be synthesized, stored, and released. Upon release, it will act on cholinergic receptors. So, what are the different types of cholinergic receptors? What are the different types of cholinergic receptors? There are two major types of cholinergic receptor, as we have said before. They are either muscarinic receptor or nicotinic receptors. Nicotinic receptors. So acetylcholine will act either on muscarinic receptor or nicotinic receptor. We'll talk first about muscarinic receptor and their properties, then we'll talk about nicotinic receptors. Muscarinic receptors, as we have said, they are mostly present in the viscera. Of course, they're present centrally and as well in the central nervous system. Also, they are present in most of the organs of the body, in the viscera. There are three types of muscarinic receptor. Actually, they have encoded 5 types, but we'll talk about the most important 3 types, M1, M2, and M3. M1 is mostly present in the neurons of the CNS and the parietal cells of the stomach. Parietal cells of the stomach, which is responsible for the proton pump stimulation, or responsible for proton secretion, parietal cells. So what will happen in this case? M1, they are linked to GQ proteins, and GQ protein, we have said, it will stimulate the phospholipase C enzyme, which will lead to formation of 2-second messenger, IP3 and DAG. Therefore, stimulation of M1 receptor will activate GQ protein, which we spoke about in previous presentation, which will stimulate membrane enzyme phospholipase C beta enzyme. Phospholipase C beta enzyme will lead to formation of 2-second messenger IP3 and DAG. This IP3 will increase in intracellular calcium, as we have said before, and this increase in intracellular calcium, if it is in neuron, leads to neuronal excitability and release of neurotransmitter, and if it is parietal cell, for example, to increase acid secretion. Okay? So this is the first type of muscarinic receptor, M1 receptor. They are present in neurons and parietal cells of the GIT, where they are linked to GQ proteins. Stimulation of M1 receptor by acetylcholine will stimulate GQ protein, which will stimulate in its turn phospholipase C beta type present on the membrane, leading formation of 2-second messenger IP3 and DAG. IP3 will increase in intracellular calcium by mechanisms we have explained before, and this increase in intracellular calcium will end up with neural excitability and release of neurotransmitter if it is a neuron, or acid secretion. This is the first type, which is M1 receptor. The second type is the M2 receptor. M2 receptor. So we said muscarinic receptor. They are either M1, which are linked to phospholipase C, will lead to formation of IP3 and DAG. The M2 receptors, they are linked to GK protein. GK protein. Protein which are G protein which are linked to potassium channel. We have explained before in previous presentation. Therefore, stimulation of acetylcholine and muscarinic M2 receptors of acetylcholine will result in opening of potassium channel. Potassium will rush out of the cell, causing hyperpolarization. We have explained that before. Therefore, opening of potassium channels. So if we just imagine this is the receptor, this is the potassium channel, and this is the GK protein. Once GK protein is stimulated, it will open potassium channel. Potassium will rush out of the cell, causing hyperpolarization. This is, we said, it is inhibition. Therefore, acetylcholine by acting on M2 receptors will lead to hyperpolarization of the membrane or inhibition. This usually takes place in the SA node, SA and AV node of the heart, and as well in the arterial tissue. So acetylcholine is inhibitory on the heart rate and the conduction through AV node, and as well it inhibits the arterial tissue contraction. This is the second type of muscarinic receptor. A third type of muscarinic receptor is known as M3 receptors. M3 receptors, they are linked to GS protein, which will stimulate the same thing, phospholipase C enzyme, and this phospholipase C enzyme will lead to formation of IP3 and DAG, and the same as M1 receptor, but different G protein. This IP3 receptor and DAG will increase the intracellular calcium, and this increase in intracellular calcium will result in either smooth muscle contraction, smooth muscle contraction, or release of glands, exocrine gland release. Like saliva, like sweat gland, etc. Okay. Therefore, M3 receptors, they are present in the sweat, present on the smooth muscles, and as well exocrine glands, where the stimulation by acetylcholine will activate GS protein, which in its turn will activate phospholipase C enzyme, which will lead to formation of 2-second messengers, DAG, diacylglycerol, and isoinositol triphosphate. IP3 will increase the intracellular calcium. If this takes place in a muscle, it will cause muscle contraction, smooth muscle contraction. If it takes place in a gland, it will cause excretion of exocrine glands, like salivary gland, like a sweat gland, etc. Okay. Now, these are the muscarinic receptor. Muscarinic receptor action produced by stimulation muscarinic receptor is a graded response. A type of response produced by muscarinic stimulation is known as a graded response. We explained that before. Graded response. What does mean graded response? There is a dose response relationship, direct dose response relationship. As we increase the dose, The response will be increased till we reach maximum. Okay? Therefore, the muscarinic response is graded. That means by increasing the dose, the response will increase till we reach a certain dose where there is no more further increase in the response. This is the first type, which is the muscarinic receptors. Second type is the nicotinic receptors. Nicotinic receptors. Therefore, cholinergic receptors, we said they are either muscarinic receptor or nicotinic receptor. A nicotinic receptor, we said they are present in the neurons or CNS. They are known as N receptors related to neurons, N, nicotinic neuron, or nicotinic muscular skeleton present in the skeletal muscle. Nicotinic receptor, as we have said before, they are directly linked to sodium channel. Sodium channel. We have explained that in previous presentation. So we have acetylcholine binding as an agonist to its receptor will cause opening of sodium channel. Sodium will rush in. Sodium ions, as we know, it is present outside the cell cytoplasm. Once an agonist binds to the receptor, it will cause opening of sodium channel. Sodium will rush in, causing depolarization of the membrane. Depolarization means, as we have said before, excitation. Okay? Therefore, nicotinic receptors that are linked directly to sodium channels, where stimulation of nicotinic receptor by acetylcholine will lead to opening of sodium channels. Sodium will rush in, causing depolarization of the membrane, exchange of the polarity of the membrane. This we have explained before as excitation. This takes place in the ganglia, therefore it causes transmission of impulses through the ganglia to the postganglionic nerve fiber, and if it does take place at the neuromuscular junction, it will cause spread of excitation through the motor end plate, which will result in the excitation contraction coupling and muscle contraction at the end. Okay, so it is excitation. What about the nicotinic effect? Nicotinic effect is not graded. It is the opposite from muscarinic effect. What does this mean? Nicotinic effect is not graded. Nicotinic response is not graded. We have said that muscarinic response is graded, but nicotinic response is not graded. What does it mean? What does it mean? It means in a small doses, not graded. It means in a small dose will produce stimulation. Small dose of acetylcholine or nicotinic agonist. Small dose will produce stimulation. Stimulation, as we have said, contraction of the muscle or transmission through the ganglia or whatever, or CNS stimulation. But in high dose, it causes block. In high dose, it causes initial stimulation. Initial stimulation. First, we will see stimulation, then followed by block. في المسلز مثلا skeletal muscle will contract, then it will be paralyzed. This block is known as depolarization block. Depolarization block. Okay? Therefore, nicotinic effect in small doses cause stimulation of the nicotinic receptor. For high doses, it causes initial stimulation followed by block. Initial stimulation followed by block. This is known as depolarization block. Why this happens? Why that high doses causes block? Why? They said that this depolarization block happens through two phases. Phase 1 and phase 2. In phase 1, they said that the membrane, when it is stimulated, it stays depolarized. What does it mean? We know that all excitable membrane, they are present as in polarized state to be stimulated. Any excitable membrane is present polarized. Polarized, as we have said, sodium is concentrated outside, potassium is concentrated inside the cell. And we said that the membrane is polarized state. When it receives stimulation, it becomes depolarized. Depolarized means excited. After depolarization, it will repolarize again. Repolarize, يحصل لها repolarization again to become polarized so that it can be excited again and so on. In the case of high dose of acetylcholine or nicotinic agonist, the membrane stays in the depolarized state. It does not repolarize. That's why it's known as sustained depolarization. Sustained depolarization. Yani, what does it mean? It means that the membrane cannot be stimulated again, because to be stimulated it must be in the polarized state. Now it is left in the sustained depolarization state, therefore it cannot be stimulated again. This is block. This is inhibition. This is block. Transmission of impulses is stopped. Okay? This is known as phase 1 block. Phase 1 block. In phase 2 block, what happens? Phase 2 block. In phase 2 block, the membrane becomes polarized again. The membrane is in state of polarization. But the nicotinic receptors are not sensitive to the agonist. So nicotinic receptor becomes insensitive to the agonist. Nicotinic insensitivity. Nicotinic receptor cannot be stimulated by the agonist, although the membrane is in polarized state. But the nicotinic receptor are not sensitive anymore to the agonist. So what will happen? A block. Block will take place. Okay? So, nicotinic response, any nicotinic agonist, in small dose, it produces stimulation by opening of, we have said, sodium channels, and sodium will rush in, causing depolarization of the membrane. In high dose, it causes initial stimulation because initially it will open the sodium channel, so it causes depolarization of the membrane. This initial stimulation is followed by a block, This block is due to two phases. Phase 1, we said, in the membrane, after it is depolarized, it remains in the depolarized state. It does not repolarize. It remains in the depolarized state. This is known as sustained depolarization. Sustained depolarization means block. A second phase, we said the membrane becomes polarized again, but the nicotinic receptors, they become insensitive to the drug, therefore it will not respond to the nicotinic agonist. Okay? So these are the cholinergic receptors. They are either muscarinic type, which are linked to G protein, or nicotinic type, which are linked to sodium channel. So we said cholinergic receptor, they are muscarinic. Muscarinic receptor, which are linked, all of them, linked to G protein, either GQ, GS, or GK protein, or these are muscarinic type or nicotinic receptor, which are linked directly to sodium channels. muscarinic receptor response is graded. As we increase the dose, we'll get more response, while the nicotinic response is not graded. The small dose will get a response, and high dose will get a response followed by block, and this block we said is through 2 phases. Okay? So these are the cholinergic receptors. Okay? Now, acetylcholine synthesized, stored, released. After its release, it will act on cholinergic receptors, whether they are muscarinic or nicotinic. Then what will happen to it? What will happen? Inactivation. Inactivation. Process of inactivation. Removal of the effect. This is known as physiological inactivation of acetylcholine. So acetylcholine is present, as we have said, in the synapse. It will act on the receptor, and then it will be deactivated. Deactivated. How it is deactivated? By metabolism. Metabolism. Therefore, acetylcholine will be metabolized in the synapse. And we have said that metabolism takes place by enzymes, as we have said before. Therefore, acetylcholine will be metabolized by specific enzyme known as acetylcholine is an ester. Of course, acetylcholine is an ester. Therefore, metabolism of acetylcholine is done by 2 different types of enzymes. One type is known as true cholinesterase. Cholinesterase. Esterase is hydrolysis of an ester, cholinesterase. Esterase hydrolyzes acetylcholine. Therefore, it is known as true cholinesterase. This type is present in the neuronal cells, and it is known as well as true cholinesterase. It is true cholinesterase. It is responsible for metabolism of acetylcholine at neurons. Okay? A second type is known as pseudocholinesterase. It's known as pseudocholinesterase. Pseudocholinesterase. Pseudocholinesterase is present in plasma. That's why it is known as plasma cholinesterase. Plasma cholinesterase or pseudocholinesterase present in plasma and responsible for metabolism of most esters in plasma. It is responsible for metabolism of esters in plasma. Okay? That's why acetylcholine, it has a very short duration of action. ما ينفعش في العلاج. ليش؟ لأن once it enters the plasma على طول it will be metabolized by cholinesterase enzymes in a few minutes. Okay? So these are the cholinergic transmission. So what we have spoke about today, we talked about cholinergic transmission. We talked about sites where acetylcholine acts as a neurotransmitter. We talked about the synthesis, the storage and the release of acetylcholine. Then we talked about the cholinergic receptors, whether they are muscarinic or nicotinic receptors. And lastly, we talked about how acetylcholine action is terminated by the process of metabolism by cholinesterase enzyme. I would like to thank you very much for your attention. Please don't forget to press like. I hope that you stay in peace and harmony. Thank you very much. Assalamu alaikum wa rahmatullahi wa barakatuh.